Welcome dear viewers to the video on a stability study in pharma industry. This video is related to common mistakes or typical blunders in the stability studies and the improvement ways. Knowingly, unknowingly, the stability study mistakes may happen, and these mistakes lead to delay in the formulation approval, product approval, and also lead to many regulatory queries. I thought to make this video to make you known about the typical common mistakes and the improvements. Stability studies are performed to study the impact of environmental condition and impact of aging onto the formulation quality. The stability studies are directly related to the safety and efficacy of the pharmaceutical formulations, and that's why these studies are very much important. Stability studies are required and are done to determine the shelf life, determine the expiry period, to have understanding about the suitable packaging system, and also to determine the best storage conditions. This video will study the typical mistakes and the improvement ways for stability studies. The common stability study problems are the inadequate or incomplete stability study design, insufficient time points for sample loading, inadequate justification of shelf life and problems in the shelf life calculation based on the data, lack of commitment for ongoing stability studies, and failure to address the OS or OOT results. There may be many other problems, but in this video, I am covering these common problems and mistakes. Be with me till the end of this video so that you will understand the problems in the stability studies. Stability studies, as I have mentioned, critical for understanding and ensuring safety and efficacy of the formulations. This presentation will highlight the mistakes and the solutions for those problems. First is incomplete or inadequate stability study design. Always have good understanding about the guidelines and the product behavior. And if the stability study design is not proper, or it is incomplete, or it is inadequate to prove the stability of the product or to demonstrate the product stability, then there is a very much big mistake. Failing to include data for all required conditions, that means long term condition, intermediate condition, and accelerated condition. Always understand the market and the product behavior. Ensure that the product is stable at some conditions or not, and in some packs or not. So it will be determined based on to the research and development phase. The improvement way for this problem is to ensure stability protocols follow ICH guidelines and the guidelines provided by USFDA, EMA, and the country specific guidelines. Always include stability data for all climatic zones relevant to your product markets. So this is the problem and solution for the inadequate stability design. Insufficient time points. So as we have talked regarding incomplete design, that means the stability design is not covering the all stability conditions. Second is insufficient time points. Always know about the guidelines. If the guidelines are not properly understood, then there may be a problem in making the insufficient time points for stability study. So problem is or mistake is providing stability data only for few time points or short durations. The solution to this problem or mistake or blunder is include data from the required storage conditions, that is 6 months for accelerated and minimum 12 months for long term condition for most of the cases. Always understand that the long term condition is different as per the climatic zones and different as per the markets. For some markets, you might see as 30 degrees Celsius 75% RH is the long-term condition, while for some other markets, long-term condition will be 25 degree and 60% RH. And for long-term conditions, always include the time points till the end of shelf life or till the end of proposed shelf life. You can also include more time points than 12 months or 24 months, so that if you have good data or if your product is passing, then you can extend the shelf life. So this is regarding the insufficient time points. Also in this, the improvement ways to ensure data represent the entire shelf life, including stability at different time intervals. Then third is inadequate justification for shelf life. So the main aim of the stability study is to determine the shelf life, and many times tentative shelf life is provided. Sometimes extrapolation is done as per ICH guidelines and the shelf life is provided or shelf life is claimed. So the problem is, or the mistake is, proposing a shelf life without sufficient data to support it. There should be always sufficient data to support your shelf life time. The way to avoid this mistake is use statistical analysis, regression analysis to justify the proposed shelf life. Include the data showing product stability over time and under relevant storage conditions. It is very much important that always you include the regression analysis, some scientific calculations as per the guidelines, some statistical calculations as per the guidelines to justify the shelf life. Then lack of commitment for ongoing stability studies. Clearly outline the plan for ongoing stability monitoring post approval. This is the way to deal with this problem. Also ensure submission that includes timelines for continued testing and data collection. I have made a separate video on stability commitment, so you can go to that video and understand this topic in a detailed manner. Always there should be a commitment for ongoing stability studies. For example, if you have filed a product with 6-month accelerated and 6-month long-term data, then there should be a commitment that the stability study will be ongoing and the results will be, or the data will be reported to the regulatory authority. Then coming to the poor data presentation. So the problem is presenting data inconsistently or without summaries, making it hard for reviewer to evaluate. See, the data of the stability studies is required to be reviewed, and based on that, the product will be getting the shelf life approved from the regulatory bodies. There should be data which is easy to review, easy to understand. So presenting data inconsistently or without some summary tables, it becomes hard to review. The solution to this problem is use clear, well-organized table to present data. Provide concise summaries to facilitate easier review and the understanding about the stability data. So in my experience, I understood that whenever you are providing stability data, provide it batch wise, pack wise, stability condition wise in a tabular form, so that the reviewer can understand it. Also, you can highlight the failing data or the data which is out of specification. You can make a summary like the product meets the specification at accelerated, intermediate and long-term condition till 12 months or till 6 months based on to the stability study condition. So like this, if you present a data, it will become easier for reviewer to review and go ahead with the review. Then failure to address out of specification results. If the data is containing any failure to the specification, like the tablets are breaking or tablets fail. For example, if the product or tablet or capsule, it may be failing in the dissolution on stability condition at accelerated condition, it is a OS. So if the OS is ignored or it is not justified, then it is a mistake. Ignoring or failing to explain OS or aberrant results is a big mistake, and it is a common mistake. So the improvement way is to provide a root cause analysis for any OS results, outline corrective actions taken to resolve the issue, and demonstrate the product reliability. For example, if you are having a capsule soft gelatin capsule formulation, if the formulation fails at 6-month accelerated condition in the dissolution testing, then understand it, justify it, like because of cross-linking the product fails in dissolution at accelerated condition, but after using the enzymes the product is passing in the dissolution test. Also provide the corrective actions that you have included enzymes in the dissolution media, and now the kappa is taken, it is included in the protocol or protocol is revised, and the next studies will be performed with enzymes if the formulation fails in stability condition. So this is a common example for failure to address out of specification results. You can also demonstrate the justification for OS, like the product is unstable at accelerated condition, but the product is passing at intermediate and long-term conditions, and the shelf life will be assigned based on the availability of complete stability data. So this is a video regarding the common mistakes in stability studies and how to address those problems. Addressing the common mistakes in stability studies are very much required, and this ensures stronger and more reliable stability data. Following ICH guidelines, US FDA guidelines, EMA guidelines, ANVISA guidelines, and other specific guidelines are required to ensure comprehensive study design and presenting clear data with strengthened regulatory submissions and product approval. Proper stability study design is key to product success and market entry. If the stability study design or any mistake is not understood, and if it is not presented in a scientific way, then you can get refused to receive from the regulatory authority, or you can get the queries. Also the product approval can get delayed. That's why stability study is a very important study to demonstrate the product stability and also to demonstrate product safety and efficacy in the pharma industry. This video was regarding typical mistakes and blunders and improvement ways for stability study in the pharma industry. I hope this video will help you to understand the topics in detailed way. Thank you for watching the video.